How does gene therapy treat autism spectrum disorders? In this presentation, we will be discussing how gene therapy works to treat autism spectrum disorders. Autism is characterized by the occurrence of impaired social interactions, restricted, preservative, and stereotypical behavior, and abnormal communication skills. Autism is known as a spectrum disorder because there is a wide variation in the type and severity of symptoms people experience. Recent studies have reported a dramatic increase in the prevalence of autism spectrum disorders, with as many as 1 in every 59 children being diagnosed with autism. Autism is a genetically driven disorder that causes abnormalities in areas of the brain that are responsible for producing normal behaviors. Gene therapy has been shown to address the cognitive dysfunction seen in autism by improving brain activity in areas such as the frontal lobe and the temporal lobe. Some advantages of gene therapy include having long-lasting clinical benefits and the potential to modify any gene of interest to treat diseases. The genetic targets in autism. The purpose of gene therapy is to target genes that are associated with this disorder by repairing the defective genes. There are several genes that have been linked to symptoms seen in autism, which result from poor connectivity in those specific brain regions. The SHANK gene 3 is considered the strongest link to autism because it is proved to be the main cause of intellectual and language deficits in this disorder. About 1% of people with autism are missing a gene called SHANK3, which is critical for brain development. The impact of gene SHANK3 in autism. This model shows the brain of a mouse with the present SHANK3 gene and another image of the brain without the gene. It was shown that the mice with the deficient gene spend more time engaging in repetitive, restricted, and stereotyped behaviors. The mice showed impaired memory when attempting to recognize and remember objects. As you can see, the mouse in the image on the right was more interested in another object rather than its own. Now we will discuss the advanced genetic modifying tool that is used to target genes. In order to target the affected genes, a protein must be present to modify the mutations. As you can see in this model, the CRISPR gene editing tool, much like gene therapy, is used to cut the DNA by using the Cas9 protein to repair the gene by producing therapeutic effects. For this procedure, the CRISPR carrying the protein Cas9 was injected into the striatum areas of both brains. The mouse in red was given the injection as a treatment for the autistic behaviors, and the mouse on the left was given the treatment as the control for this procedure. The figure on the right shows the improvement in the brain of the mouse after the treatment was given. The changes took place in the cerebral cortex. We used the fMRI to examine the changes that we've seen over time.